I forgot to. All right. So yeah, our data will be downloaded to Scratch Space, and all the output should probably go there too because those are huge. The SAM files, they contain all of the information of the FASTQ file, basically double. So they tend to be 2 to 5 times bigger in that format. So SAM is sequence alignment map. There's another version of it called BAM, which is the binary alignment map, and that's a much smaller file. It's not human readable at all. So SAM tools can convert back and forth between SAM and BAM. We're going to keep it as SAM now because we want to be able to like look at it and get to know it. The BAM file is straight up gobbledygook, one of the zeros. Other questions about assignment 7? We'll spend some time talking about the Scratch Space for sure, but we'll make sure we all have a chance. We get any troubles. Happy? You love this so much. I would like to get a little pulse on how you're feeling about what we're learning. Like, we did this at the beginning, like week 2, and everyone was hovering around 3 or 4. Happiness. How are you feeling now? We're going up, down, stay the same.
Like in terms of how much we like
Yeah, like it or your comfort level with it, your confidence working with command line, your fears for the future, having to do this for real, that stuff.
I feel like doing the assignment like made me more comfortable with like the swing and like doing a long job. I feel like I like kept being like trying to think something was not gonna work and having to like restart it all or like just like kind of waiting on it.
And that happened.
Yeah.
But it wasn't the end of the world.
No. So like, I feel like it made me more comfortable. Oh wait, it just finished.
Yeah, I'll finish, right?
So I think it started at like 25, so that's 15-20 minutes for all of that.
Just to move files to Scratch.
But I don't know, I think that like kind of helped me like demystify and also like better understanding what FASTQ is, what is like what is it. Like I think it was a really helpful like put everything together kind of thing. I think I'm curious to see where we go with.
Oh, it's getting ugly real fast.
Yeah, so I
this
think
this is why I want to like.
Yeah, I feel like I'm nervous for this, but also like again, it's like the end of class, we need time to like learn and ask questions.
Yeah, I want to slow this down to where everyone's gonna be okay moving forward, but it's not like the learning curve is gonna ramp up. It's just the complexity of the pipeline start to finish is going from we did like 1 step and then 2, and this assignment was 3 steps. Now it's going to be potentially 40 steps. We'll try to keep it simple, but you'll see how bad this can spiral. But you're going to see new software for sure. You're going to have to figure out how to use it. But that, I'm a professional at this, and that still happens constantly. It's like, oh, a brand new tool just dropped. This is very exciting. I'm going to try it out. Crap, how do I use it? Where does this fit in my pipeline? Is it actually better? Our group projects are going to be splitting up into 2 teams. One team will do a metagenome, one will do a genome. And we're just going to have raw data, and we're going to try several different pipelines to get to the same answer and see which one's best. This is called benchmarking. So getting familiar with all the options would be nice, but more getting comfortable with finding the options and the confidence to try them, I think is most important.
Could you briefly explain the difference between the 2 approaches? like,
Yeah, we've got a whole lesson on
Oh,
that
okay.
coming up. It's okay, yeah. Promise. We'll get to that. Anyone else care to share this safe space?
Yeah, I feel like what I feel like doing is
Okay.
I like that's very nice.
There's, when I think about how to teach this, there's no point in me just teaching you like, here are the tools you'll use, because they change. Constantly, every year, we're getting better software and new approaches, or a new technology comes out, like a new type of sequencing, and suddenly we've got our new tools. And so it is the case though that they all work from the command line, and they all have a help file, and they take input and output. So I wish I could say, here's how you assemble a genome, and here's the tools you use, and here's the settings. It's just not the case. So getting, if at the end of the semester you're like, I have a question, I can find the software and I can figure out how to run it on the HPC, then I'm very happy. That's the ultimate learning objective that I had in mind for this class. That's not, oh, I know how to use that and it'll be flawless. It's more like, I'm going to fuck this up 4 times in a row and then eventually get it. And I'm not afraid to fail or to feel stupid. So both important scientific skills. Anyone having less magnanimous feelings about what we're doing?
We're going to go over something further down, like for beginning to end, it was submitting, but it doesn't feel like it's really yet. It was me.
Yeah.
And I kind of was just assuming I was wrong when I was submitting it, which I was the first 2 times, but like I feel like I'm just like assuming it's gonna fail until I finally get it right. But I don't know if that'll go away.
No, it shouldn't. The assumption of failure is I think maybe a little healthy. Think of it like skepticism, like you don't trust your tools without verifying them. It's the same in the wet lab if you're pipetting. Like, you're gonna mess up and you're gonna push too hard and put too much packed glomerulus in there. Okay, well, we do all of our little checks along the way to kind of like see what's happening because who's worked in molecular at all? Notes and pipetting. You're moving around tiny drops of clear liquid for hours. You don't know what's in there. You're just like kind of trusting that you did stuff right. But along the way, you go run a gel and kind of see. It's an annoying process. It takes an hour or 2 to like check, but without that check, you have no idea where the mess up has happened. Same thing's going on here. We're moving around ones and zeros with other people's tools in a pipeline that we've stitched together. And yeah, it's gonna go wrong sometimes. And so stopping and checking and feeling that skepticism, I think is healthy. So doesn't make you a bad person. This is a good feeling that you're having. Maybe uncomfortable. You're like, meh, I'm used to it. Okay. Well, let's talk about Scratch a bit. And I will bring up our old lesson graph. Which one is that? 2. Just for this image again. Okay. A lot of arrows, but hopefully they're starting to make a bit more sense to you. we log into Bora. And it's not a huge computer, that login node for Bora. It's got 128 gigs of RAM. But we all have to share that, and so. Yeah. You've done this part now. You've sent a request to Slurm and it submitted it to the actual cluster. And the cluster can talk to Scratch 10. And so here on the login node, we're writing our scripts. We're going to say, download everything to Scratch 10. And then when our job gets submitted, it does have access. It's got a wire running. So it can talk to Scratch 10 and pull that data for us. So Scratch 10, we all have our own folder in there. Let me bring up mine.
So then, just like quick question, so like when we're submitting a SNARM job and it's like, you have like BO, like, it's like 34, so we have like, in the club, could you go back to the?
Yeah,
like,
yeah.
So we're given one of those like boxes.
Yeah.
And each of those has like the 20 cores like inside of it, is what you're saying?
Yeah.
Okay,
So there are 54 boxes in Bora, physical computers. Each one has 20 cores and 128 gigs. Right. And if you say, like in your Slurm header, nodes 1, that's saying I want one of those boxes. Bora.
would you ever request more than?
Yeah, you can. You can say I want 30 of them. But how do you run a job on 30 separate physical computers and have them talk to each other and put the results stitched back together in the right order? It's really tricky. And so IBM came up with the MPI, this massive parallel instrumentation. And if you're running like hardcore data science stuff, you're talking GPUs instead of CPUs, but you're talking to many of those at a time. And so there's a special language for computers talking to each other, physical computers talking to each other, sharing where they are in their process, and then giving each other results as needed. Most bioinformatics software doesn't do that. Because the types of algorithms that we run aren't well suited for that kind of parallel task. They're much more suited for on a single node. But on a single node, we can run up to 20 parallel processes. So for things that don't need that massive, difficult communication between clusters, you can have 20 individual processes going through the same algorithm on the same computer. And that's what those cores are.
So like hypothetically, like if you added another box, like that would make things hypothetically get faster, but or like tools aren't suited for like anything of two.
Right.
If that makes sense.
Yeah. So I know astrophysicists, astronomers and stuff that a lot of their stuff runs on 20 nodes at a time. But they're like trying to figure out black hole stuff and particle physics from way more data than we've got about mathematics. So their algorithms are specifically tailored to be able to spread across multiple machines. I have never seen a use case in bioinformatics that uses more than one node. Doesn't mean there aren't any that would come across it. Even protein prediction, like AlphaFold, that's all on one GPU box. And it's running 4 or 8 million processes at once, but they're all very tiny processes, and that's the GPU situation. So I don't know. Probably you're safe to assume that unless you go into a different branch of science, probably safe that you're going to be on one node.
And then how would you, I guess I think this was a question I had during sem 7, but how do you know like which processes can be done in parallel with like the 20 cores versus like, it's like a sequential, have to do one at a time?
You have to look
Or
along.
just, okay,
You can kind of deduce which ones are possible to do that. If it's a task that you have that's repetitive, like I need to do this exact same thing 5,000 times, that can probably be split up. And it doesn't matter what order the results come in, we're just gonna compile it all at the end.
Mhm.
And so from the start, we can just take our big data set and split it up. I need to do the same thing 20 times, so I'm gonna divide this up into 20 processes, send them out. When each finishes, they start writing to the file.
okay, so does that have to do with specified image script? Because I guess I was
Yeah.
confused for like having the for loop for, let's just like doing the mapping, and then like I had to rerun the last two, so I wrote those two separately, so I was curious if they would happen at the same. Each at the same time, because I had multiple cores, but they were sequential.
They're sequential, they're
Yeah.
serial, yeah.
Okay.
And so because you had 10 different samples, you can't split that up necessarily. But you have 10 samples, you could have written the Slurm script for each one of those and submitted all of those jobs to 10 different boxes, 10 of these different nodes. So then they could all be running at the same time. Problem with that? You gotta wait in line 10 times. You're asking for 10 of those boxes. Okay. It might be faster. You have 500 samples. Maybe you do write a Slurm script for each one of those. We can get into that later this semester, how to write a script that writes script for you. So you don't have to write 500 of them, you write one template. Yeah, but then you're just waiting in line. Your SQ is like, you got 500 things waiting there, but then they just kind of run when they run, and you may get 10 of them running at a time. And so they'll go a lot faster.
Can there ever be, sorry, this is my last
Yeah, no.
one question. Can there ever be multiple people on each of the Bora like boxes, or is it like everyone is like one at a time?
One at a time works best.
Okay.
But yeah, if you only requested 10 cores on Bora on a single node and somebody else wanted 10 cores, you could both be on there.
And does that go back to, I don't know which one it was in the Slim Code, but you could say something as like exclusive
Exclusive.
access.
Yeah, and so
Okay.
that's where I'm saying, like, don't let anybody else on here, no matter what.
So
That
otherwise.
puts you lower in line when you say that.
Mhm.
But otherwise, if you don't use all the cores and all the memory, somebody else could have their job kind of run on the same node. That leads to potential problems,
Mhm.
depending on how much memory you're using. Because the way computers work, there's like a physical location for each bit of memory. And those get assigned like an address. And two processes could want to use the same address.
Mhm.
Call that a stack overflow. So that's what the forum is named after. Yeah. Because it's like this pernicious, horrible thing. Yeah.
Okay, so like whenever we say like, I think I'm just like, things are making sense of it now, so like. When we took out like the memory thing, so by saying we want all the memory, that would probably lead them not to put anyone else on their.
It should. Yeah.
Yeah, okay.
Mhm. There are times when you don't want to request all the memory because you want some buffer, but you don't want anyone else to use it, and so then you would definitely say exclusive. And so that's what I did in my script. I wanted to leave a little buffer there for temporary storage. So in case I had an overflow, it went to a safe spot.
Mhm.
You ever run a piece of software, just normal computing, and then you have like the blue glitchy screen and it gets all like funky? That's also a stack overflow, where it's trying to store bits of your software memory in the parts of the memory that correspond to pixels on your screen. Because your screen, each pixel has a spot in your memory. And if one of your programs you're running writes something to that, it just comes out like glitchy, gross.
So,
Fun. Computers are, I hate that I know so much about that. It's not because I want to, it's because I've failed so many times. Just kind of pick this up. Yes.
For some. So. So every time, like, say in my download script, I make a data connection. Then I would be making that from scratch.
Yeah, you can.
Okay.
Mhm.
And then everything from it, like the raw, that's all the that's no scratch.
Mhm.
And then for our SRE roundtable, we should still see that we do that connection.
In assignment 7.
Okay.
Yeah. So everything you need to reproduce assignment 7 should live in assignment 7.
Yeah.
And really all you need is the scripts and that bit of info like your SRE roundtable.
Yeah. And then reference,
Reference genome and scratch 10 too, because that's your script is the one doing that.
Yeah.
Yeah.
Okay.
So we've all got our own space in scratch 10 and it's home scratch 10. So we can reference it that way in our scripts. And that'll expand, home will expand to whoever is running it. So if I put this in my script, it'll get me there, and that is my scratch 10, but that won't work for any of your accounts. Whereas this one will. Yeah, I think you can use the tilde. I've never had it not work. Yeah. I'm hesitating because like part of me thinks that sometimes the tilde doesn't expand properly.
Well, what if we do, I guess it was like the most standard if we want like the output to go to scratch, and wasn't there issues with like the home and the.
It doesn't expand up there in your Slurm script.
Yeah, so in
There's-
Slurm script, we would do this like home, that, but then like in our script, we could do that.
Yeah.
Okay.
Okay. I looked up ways to get around that, and they're pretty hacky. I don't like them. Which I guess makes sense why everybody puts absolute paths in there. It's not matter.
you want us to do maps of that.
In your Slurm header,
Yes,
because it's not running bash yet. Yeah. So it doesn't know how to expand this. So that's bash code right there.
Yeah.
Slurm can't do that.
the in the scripts, you use this thing.
Yeah, in the scripts. So in your Slurm header, your output and error streams, that needs to be an absolute path. But in your scripts, you can do it this way. All right, so I've got a bunch of junk in mine. DU is disk usage, H for human readable. Here. Well, you can see I've got a bunch of crap in there. It's still counting the number of bytes, taking the sweet time. That could never that could never live on my home directory. And it can only live here in scratch space for how long? 60 days. Right. So when I'm happy with something on scratch, the final step is to move it where? Data 10. Right. So scratch is for our running the jobs collecting our output, and then that output, you move to data 10 for long-term storage. What you don't want to be doing is having Aura work with data 10. Like you don't want to download your files there, and you don't want to like put your output there because that one actually is really slow. Data 10 is designed for long-term storage, and it's quite slow to read and write from, especially to write to it. Yeah. Okay, so I've only got 4.1 terabytes. But some of that stuff's been deleted, and that's okay, because it's just a folder. Anything important, hopefully, it's sent to data 10. I'm gonna go into lesson 8 and show you what I've got here. The basics of a pretty primitive metagenome assembly. So I've got data, and it's just got one file in it. My accessions that I'm going to be downloading. I've got a pipeline and a README in the base directory. And then I've got a scripts directory with just 7 little steps. Set up, download, quality control, assemble, annotate, coverage, and profile. Okay. Well, let's look at lesson 8. This is actually our final real lesson of the semester. That's it. After this, we're just going to be doing stuff. Okay. Yeah. No. Yeah. So if you've got a Slurm job running, it has already learned what your scripts were. It.
So it doesn't see.
Let's say you have a pipeline that's like got a bunch of steps, and my pipeline Slurm is run this one, then run that one, then run that one. If it's in the middle of downloading and I'm like, oh crap, I need to make a quick change to quality control, you can. But it's learned everything in your Slurm script. So you can't tell it, oh no, I want to do assembly before quality control. Yeah, but you could change these. And that's another nice thing about having it modular. This download for one file, the download and setup is taking like half an hour for me. Quality control will take another few hours. Assembly could take a day, 24, 36 hours. Annotation, maybe 6 to 12 hours. Coverage, 5 minutes. And then profile, a couple hours. 2 days. Or you can email the admins and say, I have a long pipeline that needs to run, and I have a reservation for 4 days. They can do that for you. So we're going to do a metagenome analysis of some sort. I'm going to let you all help decide what questions we want to answer, but could I make this smaller? It's going to kind of put together everything we've done. We're building a pipeline, a nice modular one. We're going to be sending all files and downloads to scratch, putting our output there. We're going to have all kinds of Unix tool stuff as part of our pipeline to inspect our files and look at things. We'll probably have some nice if statements in there. We'll have some for loops. Just trying to make a pipeline that not only works this time, but will work on any future files that we want to do the same process on. So let's look at the basics of the workflow. First, we've got to download the data. or access it somehow, right? If you sequence this yourself, where's that data going to be? You send off your samples to the sequencing center on dry ice, and you're all excited. And then 6 weeks later, you get an email back from them that says, "Congratulations, it's done." They probably didn't give you a file. They probably gave you an FTP link and a username and password that's good for 30 days. Okay, so to download the data, that's going to be part of that workflow, even if it's data you generated yourself. Now, if you're a crazy person, you've got your own sequencer, some people still do this. And you sequence it yourself, and now it's on the machine. Where's your data? Not on the HPC yet. You gotta hook up your hard drive to it, plug it in there, and then do what? FTP that thing straight up. If you find yourself in a position where you're generating this data and you get that FTP link, by all means, download it and then save it in 5 or 6 different spots so it doesn't go away, because the sequencing center will delete it after 60 days. So we're going to get our data, and then of course the quality control. Some of those sequences will be garbage. You've used FastP before, so we can do our quality control there. BB Tools has its own quality control software that has a lot more options we could do. But look, we've started to get some options here. Adapter handling. What is an adapter? It's the amplifying stuff. Yeah, so you've got your primers maybe, if you're doing meta-amplicon work, like your ITS1 and ITS2 primers. But also to sequence on an aluminum machine. This technology is a bit weird, but you're washing your DNA over like a film that's going to bind to individual strands of DNA in certain little spots. And then it has to amplify each little spot, but it won't bind to that chip unless it's got a certain DNA sequence adapter. And so these chips it's really magical technology, but they've got a complementary base pair that's been programmed on there, a sequence of like 18 or 20. And that's 18 to 20 that ideally is never found in nature, so it won't bind to stuff that it's not supposed to. And you attach physically an adapter sequence on the ends of your DNA fragments. So this is the library prep step of before you sequence your DNA. You've got to like ligand on adapter sequences so that Illumina can read it. But that's not real DNA. That's not part of your data. So you got to remove them. How do you know how to remove them? You know what the sequence is, and you know it's on the ends. You could do that. We could write a script that does that, but we don't have to, because FastP will take care of it. It did it in the background, you didn't even know it. It automatically looked for known adapters from a file that's present in the FastP. Okay, well, the new technology comes along and it uses different adapters. We have to take that into consideration. So we have to think about, how do we want to handle adapters? Do we want to tell it, hey, we use this special adapter this time? Quality trimming thresholds. Is the average quality score of 20 good enough for our use? Do we want to bump that to 25? Do we want to trim the ends? We need a sliding window that looks for just a drop in base pair quality in the cut there. Poly G, poly X trimming. Illumina NextSeq technology has this really annoying property that it puts poly G tails on the end of not all, but most sequences. It'd be like 20 or 100 G's in a row. That's not real. That's just part of the dark cycle of the way the technology works. So we have to keep that in mind. We want to normalize our reads.
Is that like finding the bias with like the shotgun?
Yeah. So if we take a leaf, like a hole punch, and punch a leaf of a plant, and just grind up all the DNA and sequence that, 95% of it is going to be plant DNA, right? We're interested in the bacteria that's there or viruses or something else. We don't want it swamping it out. And so if we see the same exact sequence 1,000,000 times, we can just reduce that down and say, I just want to see it 100 times. And so we can normalize our data. And so we're sequencing or we're getting more of what we want, and not the junk that's in there. But that's not automatic. So quality control is like, this is just 4 questions here, but it's actually more like 100 or so questions that we have to address. How do we want to control the data? I mean, how do you decide? You just go with the defaults?
I think it's in the context of your data, but
In context with your data and with the questions that you're trying to ask. If you want all that plant DNA because you're trying to construct the plant genome, then you don't want to do that normalization stuff.
in terms of like a shotgun versus like the Biocomputing, when you're doing like adapter and like normalizing for that kind of stuff, like how do you decide, say if like, you have like your node adapter for when you're doing like that kind of stuff, you still like is there any point to normalize them because it's like you're already like the amplicon stuff is like already setting up your data to be what you want.
Yeah, probably not. Yeah. The normalization step is, I've only seen it used in shotgun and kind of only when the assembly didn't work. People have gone back. I mean, don't get too bogged down in these like questions because every step that we see is gonna have dozens of them. All right, so we've quality control, we've gotten our data prepped. It's now somehow the way we like it, cleaned up, and then we need to assemble it. What's that mean? Here's a picture. So the different organisms are different colors represented here. And you've extracted DNA from some mixed sample, and you've got a bunch of DNA from all these organisms. That can't be sequenced with Illumina. It's got to be broken up into shorter bits. There's a lot of ways you can do this by just shaking it with beads, physically breaking it. Or we've got enzymes that'll cut it at certain restriction sites. Or you can do a sound bath, so sonicator, you can break it up. And there's a million little protocols, and you can look them up for shearing your DNA and all that. And then you get your sequences, and presto, you've got yourself a file of sequences. But all those sequences come from a bunch of unknown organisms. This is what the meta and metagenome is talking about. If you're assembling a human genome, you start with a single nucleus, right? And so there you have just one organism. We know everything in there should be human and bacteria because mitochondria are bacteria. So the assembly is taking all these short fragments and trying to turn them into longer fragments. So like reconstructing the original DNA state the best we can. How do you do that? It's all math and weird stuff. De Broglie graphs, it's graph theory. It does that. And so people doing genomics have like several graph theory classes that they take in grad school. Or you can skip it like me and just do the coding part. It's not pretty, is it? Yeah.
There's just like so much proofs.
Yeah. Well, let's see. Stop. The hell? All right. Go away, ad block. I don't want that. Let's see if we can get a nice image of dendrochronology. There we go. This is the original theory of how we reassemble short fragments into a longer picture. And it does come from dendrochronology, where we use tree rings to establish a chronological scale. The tree rings come in two flavors, good year and bad year, right? And you can like look at the history of a tree and see which years it grew more or less. But that goes for all the trees in the area. And so if you get this pattern that says like short, short, long, 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 short, short, long, 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 something recognizable, and then you see that there's another tree over here, just a fallen log, you know that tree's older, you can kind of match up and see where the rings overlap perfectly. And so we've got this one tree, it's real old, but then it died. And so we're, oops, out of luck, that's as far back as we can go, right? Well, no, because this other tree, we get the perfect matchup right there with its rings. And then this one matches up here. And so we're taking a bunch of trees in a given area, even going back into petrified trees. And so we can get exact years back for thousands and thousands of years using dendrochronology. Same thing for DNA sequences. It is astronomically unlikely that a given sequence of 150 bases will ever match up with another one if they weren't from the same organism. And so if we've got a string that's 300 bases long and 50 of them match up with the start of this other string, well, boom, those go together. They're from the same organism. And now we've got 600 bases long. Just keep doing that and we can assemble a whole genome. Well, that is computationally impossible because it has to memorize every single sequence in there in order to be able to map them up. And then it takes forever because it's got to pair every sequence with every other sequence in a sliding window. This is how we assemble with long read technology, though, because there's so many fewer reads and they're so much longer. So Oxford Nanopore, this is the way to go. For short reads, we kind of had to move away from this dendrochronology framework. Oh, I always spell it wrong. And let's not get too into this, but it's worth looking at because this is what you're about to do. It breaks your DNA up into k-mers, k meaning some number. And we start in all examples with 3. And so if you've got a string of AAT, TTT, you've got a k-mer that's AAA, then you've got one that's AAT, then you've got one that's ATT, and then TTT. And so it breaks it up into all the possible 3-letter long words that come out of that string. Okay, so that's what we're seeing here. And then we use graph math to put together all the possible combinations of where they go together. Pretty exciting, right? That's what we're going to be doing in lesson 8. So we've had 5 days of graph theory.
Yeah,
That you should be ready to go.
I think I got the gist of it by that, that's how.
Everyone else, how much graph theory? Nothing. Great, okay. This
Yeah.
picture.
Yeah.
Okay. Well, fortunately, you know, the smart people have put together some software for us. There we go. In the assembly, spades and mega hit do all that in the background. You just need to be aware of they're doing a thing called graph assembly. So you know how to Google this, basically. And because one of those things that you're gonna have to make a decision on is the k-mer sizes that will affect your assembly. And so by default, spades is going to start with 27 mers. So it's going to do every possible word that's 27 letters long. And then it'll do 33 and then 55, and it can go up to 127. Looking for longer and longer words, trying to make a better and better assembly. So we can use spades to do that assembly process. And that's going to be nice. But that's just turning the short fragments into hopefully longer things that we call contigs, contiguous pieces of DNA. All right. For some analyses, you're done at that point. You've got your contigs. What's the shortest one you want to allow?
This becomes, let's go with 2K on that basis.
2K to 5K? Is that long enough for a full gene? For a virus, yeah. For a eukaryotic gene, with introns and exons and all that, alternative splicing sites, no, 5K to 10K for like a small gene. So we have to start thinking about like how long is long enough for us to answer our question. It depends on the question. But there we have it. If we get some that are only 500 long, maybe that actually helps us answer our question, because I do a lot of fungal work. The ITS 28S region, that's under 500 usually. So I could get like a good phylogenetic placement of that thing just from a short contig. And it's a lot better than I could have gotten from the 150 bases that I started with. So I've added information. So maybe that's good enough, but that's just another one of those questions that we're gonna have to like address. The minimum contig length, the k-mer sizes. Do we want to do error correction step? Well, yeah, probably, but also that takes a good full day per sample. But maybe we just skip it and allow a bit more error. Okay, we've assembled our contigs. So we've got long pieces of DNA that have been reassembled. They may have taxonomic information. They may have like a gene with like a start and stop code on. We can say this is a gene. What can we do now?
Blasting.
Okay, so yeah, we could blast those.
We need to find the
What's an ORF?
open.
Open reading frame. Yeah, so we need to find that start and stop, and we need to like say this is the frame in which the amino acids are being ordered, and we've got ourselves a gene. So yeah, we need to detect the ORFs, the open reading frames, identify the genes on there, whether they're partial or complete and all that. This is all annotation. So some of those big sequences of DNA, those contigs that we built, will have nothing useful. It'll just be tandem repeat stuff. Imagine all the junk DNA that's in the human genome. A big percent of your genome is just telomeres. What use is that? Doing genes on there. It actually can be useful for some questions. Telomere length. is like an important biomarker for some diseases and stuff. And so it depends on your question. But we're going to annotate this, and annotation can detect the telomere, but also we're just essentially most of the time looking for genes. So we need to blast those or some other algorithm to kind of see what they are. So yeah, what database we're going to use. Are we just going to align them to the human reference genome, maybe? We're doing metagenome, that human reference genome is going to be useless. NR. So the NCBI NR is the non-redundant protein database. It is huge and full of garbage. Okay. Anybody can upload anything to NCBI. It makes it into the NR. You can annotate it incorrectly. It's full of, it's not curated.
I thought they were doing.
No, they make it non-redundant.
What's the difference between, what is that?
They took out all the repetition, so it's a smaller file. It's still huge. It's very costly to blast against the NR, because every single contig has to go against the humongous database. And so, yeah, I do that because I'm often looking for like undescribed proteins that aren't in the regular databases, but we're doing human like gut sample, which is what we're going to be doing a lot easier, and we've got good databases for that. And so Uniprot, we've got Silva, we've got just a number of databases. Some are like geared towards human samples, some are geared towards mouse samples. These are curated by experts who have like found this protein. It's been isolated. They've done the southern blot on it, like they pulled it out with antibodies. They know what this protein more or less does. So it's got like peer-reviewed study behind it. It's got a name, and then here's what that protein looks like. So when we match our open reading frames against that database and it finds a hit, probably going to get like a name and a protein family. Annotation isn't a perfect thing, but it's probably going to give you something like, hey, this gene looks an awful lot like this thing in the database. Not exactly, but we can be more or less confident that your gene has something to do with carbon cycle. Or this sure looks like NIFH, or nitrogen fixation. So we can kind of get a handle on what the genes in our sample can do. That's kind of cool because we can build a profile of like all the metabolic activity that's possible in our sample. So from all the mix of microbes and eukaryotes and viruses in there, we can get like a metabolic profile. Not what it is doing, because that's RNA, mRNA, but what it can do, the potential in its genome. So annotation. Here's my favorite, the KEGG, Kyoto Encyclopedia of Genes and Genomes.
Is this one still a thing?
Yeah, it's actually a paid service.
Is there a free one that's not out there?
Yeah, I think so. This one's really nice. It links up fairly well with Uniprot and all the others, but for example, we've got metabolism. Oh, look. All these genes have been like classified and nested within each other. So we can kind of explore here. Carbon metabolism. Whew. All right. Recognize any of these? Listen. All right, what have we got here? Some stuff that you may have seen before. Maybe. Yeah, maybe plus ATP, glycerate. Anything? Glucose, fructose, ribose. CO2. Yeah. What is this? This is just carbon metabolism. All right, here's glycolysis. Let's just highlight that one.
So this is just showing us the different pathways.
Yeah, these are the pathways. Okay, so I wanted to show you the pathways because look, we start with glycolysis. You've heard of this, right? So you've got glucose right here, and there's a gene that can act on glucose and turn it into glucose 6B. And there's a gene that can act on that, turn it into fructose 6B. Oh, so these are all the enzymatic transformations of one molecule into another. Well, we know that that's what genes do. They make proteins, mostly. Let's stick with the easy biology version, where a gene encodes for a protein which has some service out there as an enzyme. And each protein can do one or more things, but it's usually like, okay, if I need to go from fructose 1,6-P2 to glyceraldehyde 3P, I need this pathway right here.
And is that gene like the specific to humans, to animals?
Most things, because we inherited this pathway from our bacterial ancestors, and that's actually where it's taking place. It's in our mitochondria, so it's still bacteria. Depends on the gene. Okay, so we've got this compound. Here's the reaction. And here's the pathways that it's found in. Here's the different modules that it's found in, and here's the enzymes that can work on it. Okay, glycerol 3-phosphate 1-dehydrogenase. Boom. Well, where do we get that? From a gene. So it goes all the way down. It's all this knowledge and like boring cell bio stuff and biochemistry has a purpose because these genes, we can map our reads to them and it can basically say like, look, I think this gene serves this reaction. And if it can't say that exactly, it can say it looks a lot like a gene that serves this reaction. So the odds that it does something totally different are pretty low. So that's what annotation can be all about, is we're trying to assign functions to our open reading frames. Okay, and then the next step in our pipeline, estimating the coverage, or another way of saying that is like, how much of this gene is present in your sample? Why do you care? If it's like super highly expressed or expressed as the wrong word, represented in a given metagenomic sample, that means there's probably a lot of that organism there doing a lot of that process. If it's really tiny, it's probably not environmentally important. So we've got our long reads, but it's just like, hey, here's this content we found. On there, we found a gene. We know that it links up to this process. Then how do you determine how much of that was in your original sample? We've gone through a whole lot of steps here. Right? Right now, we just have a long stretch of DNA. How much of it was there? We have to go back to our raw data or like quality control data and map it against our context. How many times did a stretch of DNA map to that gene? That gives you an estimate of how abundant that gene was in the original sample. And you guys have done that step.
That's the same.
Yeah, that's the SAM file. So we use BBMap. That's usually what I use. There's other tools in there, Minimap. And there's a lot of questions there. How do you handle ambiguous mapping? Okay, I've got a stretch of DNA here in the raw reads that maps to 2 possible spots on your config. Which one do I assign it to? We got our config stretched out, nice and straight. We've got a short little read, and it maps equally well to 2 spots in that config.
Oh, is it a
Which one should you map it to?
circular?
This isn't circularized yet.
They not usually both.
You get to decide. Like, how do we handle that? To the first one, you assign it randomly, you map it to both. You map it to both and then count it as half of a map to each. If you map it to both and count it as 1, thereby biasing things. Right, so we have to think about that. We have to think about how good of a mapping do we want for it to count. We want to filter out things that map with the forward but not the reverse, right? Decisions. But that gives us our like coverage estimate or kind of the abundance estimate for each gene in our sample.
If it's like the same gene that's like getting mapped, why does it matter in the context of both of them?
We don't know because, okay, you can have 2 genes that are functionally identical for the first 100 bases, and then they have different amino acid sequences on down, and this one maps to the start of both those genes. We don't know which one it goes to. Yeah. So we could also decide to throw it out if it's not mapping unambiguously. So get rid of it. That's the conservative thing to do. But then you're kind of losing some of that coverage information.
Is that usually in the annotation stuff?
Yeah, or in the coverage estimation stuff. So after we've annotated.
And then you take the data from the annotation and then you just map that back to SAM file.
Yeah. Are y'all confused yet? You memorized all these different options? Frantically typing? Okay. And then we can do another thing, taxonomic assignment. So, yeah. So we can do that from our short reads. Maybe 150 bases is long enough to get taxonomy. For fungi, that's often the case. For bacteria, you can get at least genus from that usually. But maybe that 150 bases isn't in a taxonomically informing area of the genome, and it's just actually a telomere and gives you nothing. So we assemble our contigs, we can assign taxonomy to the contigs as well. And so then we can say, not only do we have this gene, and it does nitrogen fixation, but it comes from this species. Because there are more than one species that can do nitrogen fixation. So we're starting to get this profile of our entire sample. What started as clear liquid in a tube has gone through all kinds of horrible, confusing things, and we've made a lot of choices along the way, all of which affect the outcome. And now we've got a profile of the functions that community could do and who was there. All right, we can take this another step, and I'm not going to do this with us. Not yet during class. But we can take all those contigs and make a determination of which contigs belong together. This is kind of a weird thing. It feels like smoke and mirrors to me, but they do stuff like GC percentages and conserved motifs, and it's machine learning that goes in there. It's looking for patterns, and it trains itself on these contigs probably belong in the same genome. So that's binning. We can take these contigs and we can bin some of them into this putative genome and some of them into this putative genome. And then we can reassemble each of those into ideally whole genomes. So in other words, we've come full circle, and where we started with full genomes up here, they've been chopped up, reassembled, binned, and we try to assemble full genomes. In the case of bacteria, circularized full genomes. We call those MAGs, metagenome assembled genomes.
Is there like the right assumption that like most of the time, like it's not big?
Yeah, almost never. Yeah.
So you're just
Sometimes you can get some.
how do you like then like when you get to like that final step like from when it gets like the thing,
How do you know if it's complete?
or how do you know like that this is like actually what you're saying it is because like are like the genes like super like common between bacteria and plants and viruses and like
Yeah.
how do you like get like the final assumption saying like I did this whole thing and like now I know what it is, but I feel like all the stats we're getting is like
Yeah.
all the
So
things and, like,
I don't have a marker.
I got
Oh.
one.
This one. Okay. I think I'm good. Okay, I got two colors. So let's say we've got a contig, just this nice long stretch of DNA. We assembled that, it's nice and happy, and somewhere in there we found an open reading frame that we're pretty sure is a gene, but it's a super common gene, like alcohol dehydrogenase. So you and yeast have that exact same gene. Okay. So how do we know if that goes to a yeast or you? The flanking regions, that other stuff, this is not coding, and so it's freer to mutate, and so it gets little mutations that will look different from the yeast version. And that's how we're telling them apart. And so when we assign taxonomy to this whole stretch, we can say this contig goes to humans. We recognize the human pattern here, and we found alcohol dehydrogenase.
so like the fast Q would be like the red thing, and then
No, the fast
is
Q,
map, or-
this would be the contig that's
Yeah.
been assembled out of the tiny pieces. So that long bar.
uh-huh.
All the short reads underneath it are showing how we kind of like assembled the contig here. Okay. And then we find our little genes in that. And so, yeah, there's going to be more than one gene on your contig if it's nice and long. And so from the context of the things around the genes, we can tell who it belongs to.
And like when like if they're being like blasted or like annotated, like that context or those mutation contexts are already in the database, like those are like already a part of it.
Hey, I hope so. If not, then guess what? We don't know who it belongs to. This is my personal hell. Because I study unstudied areas, right? Deep soil, ocean, fungi, they're not in the databases. And so often what I get is nothing. Like, hey, I found all these genes, I don't know who they belong to. Or I found all these genes, I can tell they're genes, but I don't even know what they do. These are new to science. Oh, cool. But useless to me. And so the contig itself may have many actual genes on it. And we're annotating those. When we assign taxonomy, this whole thing gets taken into account. Well, this gets out of hand because we have to make choices at every step. I did a calculation just looking at all the options that you can pick for these steps here. And it came out to 33,554,432 possible answers. That's every possible combination of those decisions. And I made it easy by just saying there's only 2 options for each. You could do quality equals 21, quality equals 25, quality equals 24. This is an exponentially horrible problem. So what do you do? And that's where we're gonna start looking. Okay. So Thursday after the quiz, we're gonna start looking at my setup and download scripts, and then we're gonna do this one sample. Just the one. You can go ahead and get that set up. Put it inside of data accessions.txt, and then take a look at it. It's from this one study. It's human gut. Pick that because we're likely to get some metagenome assembled genomes. We'll get some annotations that make sense. But look at these questions and think, where do I want this to go? I don't care, but I'm gonna let you guys decide. And based on the questions that we want to answer, we'll see how we go in and kind of make those choices for each step of the pipeline. If you're feeling very confused, that's fine. You don't have to memorize all this. This picture here is good enough. We had whole genomes, we chopped them up, we tried to put them back together, we tried to find the genes on the longer pieces, and then we try to find which long pieces go together in the same genome. That help you understand what a metagenome is? Slightly. Okay. Slightly is good.
I mean, better than no knowledge, but.
Yeah, no. You'll get to know one firsthand. And then you'll get to know many of them. And the team who gets the genome, the metagenome, won't have it any easier. It's got its own problems. I am going to make it a eukaryote. Right.